Good morning. Today we will be starting sex determination in humans. Now as you can see the left side of the picture, there is a male and a female. And in female, in case of humans, there are two X chromosomes. And in case of males, there is one X chromosome and one Y chromosome. So what happens? A single oocyte, an oocyte has a single X chromosome, and the sperm has either an X chromosome or a Y chromosome. If a Y-bearing sperm fertilizes an oocyte, the zygote is a male zygote, and if an X-bearing sperm fertilizes the oocyte, the zygote is a female zygote. So it is evident that in this case, specifically in case of females, the presence of this Y chromosome is actually contributing to the decision of whether the individual will be a male or a female. So the Y chromosome is contributing into this decision. Now, when we talk about sexual identities, they occur at various levels. So if we start with the levels, so there is a chromosomal component where XY male, XX female. So this decision that whether the zygote will be a male or a female depends on the chromosomal composition and occurs at the time of fertilization. Coming to gonadal sex, 6 weeks after fertilization, the undifferentiated structures begins to develop as testes or as ovaries. Now, here I'd like to point out one thing, that the default fate, that is after 6 to 8 weeks of development, the default fate of an embryo is female unless and until a specific gene in the Y chromosome becomes activated and produces a protein. So I'll be going through it in the later slides. Phenotypic sex. Phenotypically, the sexual characteristics starts after 8 weeks of fertilization, and final, the secondary characteristics totally develops during the time of puberty in response to the male and the female hormones. Gender identity, usually if it's a male, will have a strong attraction for a female. This is the usual gender identity, and sexual orientation attracting to same or opposite sex. Gender identity is that it is going to have a self-identity, strong feelings of being male or strong feelings of having a female developmental thought process. Now, this is another view of the X chromosome, and this is the Y chromosome. As you can see, in comparison with size, if you can compare the size, the Y chromosome is comparatively very small. Now, in case of female, as I'm reiterating, it is 2 X chromosomes, and in case of male, it is a X and a Y chromosome. When you talk about sex chromosomes in male and female, the sex with 2 different X chromosomes, the sex with two different sex chromosomes, one is the X and the other is the Y, is known as heterogametic sex, and the other with the same X chromosomes are known as homogametic sex. In human, males are heterogametic, while in case of females, they are homogametic. While if we take this case of some other species such as birds and snakes, in birds it has been seen that the males are homogametic and they are ZZ, and the females are heterogametic, they are ZW. Now, as I started saying with this slide that the size is different, the human X chromosome has 1500 genes, and as you can compare the size and is much larger than the Y chromosome that has 231 genes. In meiosis, in the male, X and a Y chromosome act as if they are a pair of homologs. So during meiosis, the X and the Y pair as if homologs, even though there is very little similarity of the genetic composition between the X chromosome and the Y chromosome. Now moving on to the next slide. So this is the female genotype, this is the male genotype. So as I said, in absence of the Y chromosome, the default fate of the embryo is to develop into an ovary. The embryonal gonadal cortex actually develops into the ovary, and finally female sexual characteristics develop. But in case of presence of the Y chromosome, as you can see in the Y chromosome, there is a yellow block. This yellow block is known as the SRY gene. The SRY gene produces a specific transcription factor which is known as the testis determining factor. Now, under the presence of the testis determining factor, the medulla is induced to develop, the medulla of the embryonic gonads, the medulla of the embryonic gonads is induced to develop into testes. And finally, the testis produce testosterone, a hormone that actually initiates male sexual development. If we look into this picture, so this is the stage of undifferentiated gonad, where we are having both the embryonal gonads have both the male and the female counterparts. The red tube is actually the female structure, while the blue one is actually the male structure. The red tube is the Mullerian duct and the blue one is the Wolffian duct, and these are the embryonal gonads. So in presence of the SRY gene, as I showed here, the SRY gene is capable of producing the testis determining factor. So in presence of the SRY gene, the testes develop, the medulla of the embryonic gonads is induced to develop into testes, while the female structures degenerate. As you can see, the Mullerian duct is degenerating, and finally the testes develop, that is the male gonadal structures development is completed. But in this case, where there is the SRY gene is absent, as you can see, the embryonic gonads develop into the female structures, where automatically the Wolffian duct is degenerated and the female structures. So it is evident that if an embryo has, the default fate of the embryo is to develop into a female structure. It is only the presence of the Y chromosome that can initiate this developmental process. So, in presence of the Y chromosome and the SRY gene present on the Y chromosome, actually the development of the testes occurs, and we get the development of the male characteristics. Now, let's look into a bit detail into the Y chromosome. So this is the Y chromosome. So as you can see, there is a small arm and there is a large arm. And we have a centromere. At the extreme end, if you can see, both in case of the small and the large arm, at the extreme end, you can see that there is a structure written as PAR1. So PAR is pseudo-autosomal region. And there is a large central part which is written as MSY, that is the non-recombining region or the male specific region of Y chromosome, while PAR is the, as I said, the pseudo-autosomal region. So this portion can combine with the X chromosome during meiosis and helps in the detachment and helps in the spindle formation, and as a result the meiosis process can initiate. Now if we look into the MSY region, or now let's look into this region in a bit detail. So as I said, present on both the ends of the Y chromosome are the pseudo-autosomal region, and they share homology with the regions of the X chromosome, and synapse and recombine with it during meiosis. The presence of the pseudo-autosomal, the yellow color here, is very important because the pairing of this region is critical for segregation of the X and the Y chromosome during male gametogenesis. The remainder of the chromosome, or here, the remainder of the chromosome, about 95% of it does not synapse or recombine with the X chromosome. Since this portion does not synapse or recombines with the X chromosome, This portion has been named as the non-recombining region of Y. So it was initially coined as NRY, but recently currently it has been known as the male specific region of the Y, or the MSY. If we look into this particular structure, they see the MSY has different shades. So there are shades of blue and there are shades of gray. The blue shades actually indicate those portions which are euchromatic. So you have an idea of what is a euchromatin, where the DNA becomes accessible to the to the RNA transcription machinery, and RNA synthesis can take place. So basically, the euchromatin region are those regions where genes are present and are active. And the gray regions are the heterochromatin region, where the DNA is tightly wound around the nucleosome, as a result of which the DNA is not accessible to the elements of the transcription machinery. And usually in most cases, the heterochromatic region consists of repetitive sequences. So if we look into this particular picture where there are two shades of color, it can be again said that the MSY is divided equally between euchromatic region containing functional genes and heterochromatic region lacking genes. Within euchromatin and adjacent to the PAR, so this is the pseudo-autosomal region, and adjacent to the PAR of the short arm of the Y chromosome is a critical gene that controls the male sex development, called the sex determining region Y, SRY. So this is the location of SRY, sitting in the short arm just adjacent to PAR. In humans, the absence of a Y chromosome, almost as I again and again said, leads to a female development. That is, this gene is absent from the X chromosome. So SRY does not have a counterpart in the X chromosome. Now, if we look into this very simplified picture, as you can see, there is a specific gene which is present in the Y chromosome, and there is a gene which is present in the autosome. Now, we have to keep this in mind that the total development of male baby depends obviously, the cascade starts with SRY, but there are also certain autosomes involved in the process. Now SRY, as you can see, produces the testis determining factor, and there is a gene here written here, TMF, which produces the testosterone receptor. So these two genes results in the testosterone receptor complex, signals male differentiation, male secondary sexual characteristics develop. So this is basically the very schematic pathway of the development of the secondary sexual characters. Now coming to the SRY, a bit more detailed. At 6 to 8 weeks of development, the SRY gene becomes active in XY embryos. The SRY, as you can see, is encoding a protein that causes the undifferential gonadal tissues I showed you earlier of the embryo to form the testes. This protein is called testis determining factor. Now, as I had said in the earlier slide, the testis determining factor is believed to function as a transcription factor. So this is actually a transcription factor. It's a DNA binding protein that interacts directly with regulatory sequences of other genes to stimulate their expression. I just now said in the X chromosome there is this gene TMF, but apart from that, I said that some autosomal genes are also important and play a big role in this cascade, that is initiated by the SRY gene. Two famous autosomal genes. One is the human SOX9 gene, which when activated by SRY, so this autosomal gene is also the target of SRY. So when SOX9 is activated by SRY, it leads to the differentiation of the cells that forms the seminiferous tubules that contains the male germ cells. So this is one gene. Let me see here. So as you can see, this was the Mullerian duct, this was the Wolffian duct, and when the SRY gene is activated, so apart there are a few other genes, autosomal genes, that are also roped in, and one is, as I just now said, SOX9, which when activated leads to the differentiation of the cells that form the seminiferous tubules. Another potential target of the testis determining factor is the gene for Mullerian inhibiting substance. So this is the Mullerian duct. So this is the female Mullerian duct. So if SRY gene is activated, automatically the female Mullerian duct needs to be degenerated. So another target of the SRY gene is the gene for Mullerian inhibiting substance, also called Mullerian inhibiting hormone or anti-Mullerian. Now, cells of the developing testes. So when the SRY gene is activated and the process of testis development has started, the cells of the developing testes secrete this Mullerian inhibiting substance. The Mullerian inhibiting protein causes regression, as you can see, the red hashed dots. So the Mullerian inhibiting substance or the protein causes regression, atrophy of the cells in the Mullerian duct. Degeneration of the duct, as you can see, degeneration of the duct prevents formation of the male reproductive tract. Very sorry. Degeneration of the duct prevents formation of the female reproductive tract. So, the Mullerian inhibiting substance produced by the developing testis cell actually results in the degeneration of this female Mullerian duct. So, apart from the SRY gene playing a vital role in initiating the cascade of development of the embryonic gonads towards testes, as I just now said, there are 2 or 3 other very vital autosomal genes which also play an important function. One is an SOX9 gene, and the other is the gene producing the Mullerian inhibiting substance. Sorry. So, coming to summarize the sexual development, the stages of sexual development. As you can see, the SRY gene is on, development of testes, which results as soon as the testis starts developing. We just now talked about this. The female structures degenerate. The testes and in presence of the SRY gene, the testes produce the testosterone, which is actually produced from cholesterol. So there is a testosterone, and the testosterone passes through the plasma membrane, and as you can see, there are receptors. So they bind with the receptors, resulting in the internal development of the internal male structure development, like epididymis, ductus differentia, seminal vesicle, ejaculatory duct. So once the testosterone is produced, it passes through the plasma membrane, binds with the receptors, resulting in the development of the internal male structures. From the testosterone, further the dihydrotestosterone is produced, which passes through the plasma membrane, resulting in the development of the external male structures. Now, there are the red colors. The red colors, as you can see, the red colors are actually diseases that are indicated by red. So, if there is any disease in the receptor path, in the development of the receptors, any problem in the differentiation of the testosterone to dihydrotestosterone, or if there is any problem of the receptors that will accept the dihydrotestosterone, we usually get these three kind of diseases. And if there is any defect in this pathway, that is congenital adrenal hyperplasia. So in summary, this is actually how the Y chromosome initiates the cascade of male sex determination in case of humans. So the default fate, again I'm repeating, of the embryonic gonads is to become a female. It is presence of the Y chromosome, and specifically the presence of the SRY gene on the Y chromosome that sets on or starts these cascades, which ultimately leads to the development of either the internal male structures or the external male structures. Now a little bit more about the Y chromosome. I just wanted to show. So this is the Y chromosome. So as you can see, this is the P arm and this is the Q arm. The P arm is this is the male specific region. And as you can see, there are three regions here mentioned. If you look, so this is the centromere. So this is the small arm. This is the large arm. Okay. So this is the centromere, and when it has been studied, the sequences of Y chromosome has been studied. So this was a work done by Skaletsky in 2003. So it has been seen that the MSY, now this MSY has been zoomed out. This male specific region has been zoomed out, and if you look into the colors, let us identify the colors first. So this color, the cream peach color, that is the X transpose region. Then we have this yellow colors, this is the X degenerate region, and we have a shade of blue, that is the Ampliconic region. The shaded colors are the heterochromatic region, and the green color, initially there is a green color shown here, is the pseudo-autosomal region, and this is a gray color, this is other. Now as I said, so the MSY region, the MSY consists of 23 million base pairs and can be divided into three regions as shown in the picture. The first is the X transpose region. Concentrate on the peach color. So this is the X transpose region. So now it comprises about 15% of the MSY and was originally derived from the X chromosome in course of human evolution about around 3 to 4 million years ago. The X transpose region is 99% identical to the region XQ21 of the modern human X chromosome. Two genes, both with X chromosome homologues, are present in this region. So this shows the maximum similarity with the X chromosome, the peach color. Now coming to the yellow colors. The second area of the MSY is the X degenerative region. It comprises about 20% of the MSY. This region contains DNA sequences that are even more distantly related to those present on the X chromosome, the yellow ones. The X degenerative region contains 27 single copy genes and a number of pseudogenes. So what are pseudogenes? Genes whose sequences, they were actually originated from the X chromosome, but the sequences have become degenerated sufficiently during evolution to render them non-functional. For example, the promoter element might be missing. So these are pseudogenes. And finally, there is the third area. This is the blue color area, which is the Ampliconic region. It contains about 30% of the MSY, including most of the genes closely associated with the development of testes. These genes lack counterparts on the X chromosome. So they do not have, mind it, they do not have any counterpart on the X chromosome, and their expression is limited to the testes. There are 60 transcription units divided among 9 gene families in this region, most represented by multiple copies. So there are 60 transcription units, that is 60 genes involved. Members of each family have nearly identical DNA sequences. Genes in the Ampliconic region encode proteins specific to the development and function of testes, and the products of many of these genes are directly related to fertility in males. So this is about the Y chromosome of the males. So with this, we come with the end of the topic, sex determination in humans.